Okay, let's dive right into this explainer and zoom all the way down to the microscopic building blocks of the human body. Welcome! Today we're undertaking a really incredible journey. We're going to look way past the gross organs and bones you're probably already familiar with and focus on the hidden, microscopic fabric that makes human life possible. I'm talking about your tissues. It's this fascinating world where form perfectly meets function, and honestly, understanding it completely changes how you view your own body.
Here is our roadmap for today's explainer. We'll be covering Beyond the Naked Eye, Introduction to Histology, the 4 tissue types, the art of tissue prep, and finally, the future of microscopic anatomy.
Section 1. Beyond the Naked Eye, Framing the Scale of Human Anatomy.
You know, the word anatomy actually comes from Greek roots meaning to cut apart. Historically, this mostly meant gross anatomy. Gross anatomy deals with the macroscopic stuff. It's the study of the heart, the brain, the femur, those large, easily visible structures you can observe with the naked eye. But as our technology leveled up, so did our line of sight. And that brings us to microscopic anatomy. This is the study of minute structures that absolutely require magnification. Think of gross anatomy like looking at the architecture of a whole house, right? While microscopic anatomy is pulling out a magnifying glass to examine the very grain of the wood and the specific composition of the bricks.
And that analogy brilliantly sets up the 2 main pillars of microscopic anatomy. Once you look through a microscope lens, the world divides once again. On one side, you have cytology. That's a specialized study of individual cells and all their internal components. On the other side, you have histology, which is what we're focusing on today. This is the study of how those individual cells actually team up, how they organize, and how they integrate with extracellular materials to form biological tissues.
Section 2. Introduction to Histology, Bridging Cells and Organs.
So now that we're zoomed in, let's define exactly what histology actually is. The crucial point to remember here is that histology acts as the bridge between cellular biology and gross anatomy. You have cells, right? And those cells group together to form tissues, which in turn group together to form the organs of your gross anatomy. Histology is the study of that vital middle step. It's essentially the material science of the human body. By looking at the microscopic arrangement of a tissue, anatomists can literally predict exactly what it does.
It's actually wild to trace this back. The whole field really kicked off back in the 17th century when an Italian scientist named Marcello Malpighi started using early microscopes to study the organs of bats and frogs. He was the guy who discovered those tiny hair-like capillaries and lungs, finally proving how oxygen enters the bloodstream. Then, jump forward to 1801. A French anatomist, Xavier Bichat, formally introduces the concept of a tissue. He actually described 21 distinct human tissues. And finally, in 1819, Carl Meyer officially coins the term histology, cementing it as a distinct scientific discipline.
Section 3. The 4 tissue types, the materials of the human machine.
Despite the absolute mind-boggling complexity of the human organism, it all boils down to just 4 basic materials. Every single organ in your body, from your brain to your skin to your stomach, is constructed from some combination of these 4 basic tissue types: epithelial, connective, muscle, and nervous tissue. Each one of these has a highly specific microscopic architecture designed to do a highly specific job. Let's break them down one by one.
First up is epithelial tissue. Think of this as your body's boundary layer. It covers all your body's surfaces, lines your internal cavities, and even forms your glands. And form dictates function perfectly here. Histologists classify epithelium by the shape of the cells and how many layers there are. For instance, a simple squamous epithelium is a single, incredibly thin layer of flat cells. That's absolutely perfect for the lungs where you need really rapid gas exchange. But for your outer skin, you have stratified squamous epithelium. That's multiple stacked layers of cells designed to take a beating and provide a really robust protective barrier. Next up is connective tissue. This is actually the most abundant and widely distributed tissue in your entire body. It provides support, protection, and structural integration. Now, what's truly fascinating here is the sheer variety. When we say connective tissue, you probably immediately think of dense tendons and ligaments, or maybe the hard, mineralized matrix of your bones. But did you know blood and lymph are also classified as connective tissues? No way, right? They just happen to have a liquid extracellular matrix instead of a solid one, but they all belong to the exact same tissue family.
Our third category is muscle tissue, which is highly specialized for contraction and generating force. Histologists categorize muscle based on 2 main things: its microscopic appearance, specifically whether it has a striped or striated pattern, and whether we consciously control it. So, skeletal muscle, which moves our bones, is striated and voluntary. Cardiac muscle in the heart is striated but involuntary. You don't have to consciously think to make your heart beat, thank goodness. And smooth muscle, which lines your blood vessels and digestive tract, is completely non-striated and involuntary.
Now, if we zoom even closer into that striated muscle fiber, we find a microscopic structure called the sarcomere. This is the ultimate functional unit of the muscle. Inside the sarcomere are these overlapping protein filaments, thin ones called actin and thick ones called myosin. According to the sliding filament theory, when a muscle contracts, these filaments physically slide past one another, literally shortening the sarcomere. Multiply that tiny microscopic sliding action by millions, and you get the macroscopic movement of lifting your arm, just a perfect example of microscopic structure dictating macroscopic movement.
The fourth and final tissue type is nervous tissue. This serves as the body's rapid response command center, integrating the entire system. It makes up your brain, your spinal cord, and all your peripheral nerves. Under the microscope, you'll see two main types of cells here. First, the star players called neurons. These are the ones actually transmitting the high-speed electrical impulses and chemical signals. And second, the neuroglia, which are these crucial supporting cells that insulate and protect the neurons. They're basically the unsung heroes of the nervous system.
Section 4. The art of tissue prep. How scientists actually see tissues.
So, you might be wondering, how do we actually get these soft, squishy structures onto a tiny glass slide to look at them? I mean, tissues are mostly water. You can't just throw them under a lens and hope for the best. Getting a piece of tissue ready for the microscope is a highly sequential pipeline, and honestly, it's a vital, delicate art form. Step 1 is fixation. Scientists use chemicals like formalin to permanently cross-link the proteins. This basically freezes the tissue in time and stops decay. Step 2 is embedding. Because tissues are full of water, they have to be chemically dehydrated with alcohol and then infiltrated with melted paraffin wax. When that wax cools down, it creates a hard, solid block containing the tissue, making it sturdy enough to actually cut. And once you have that wax block, you move to step 3, sectioning. This is done using a specialized, highly precise instrument called a microtome. To give you an idea of the scale we're talking about, the microtome cuts slices that are typically only 5 to 15 micrometers thick. For context, that's roughly 1/10 the thickness of a single human hair. It has to be cut that unimaginably thin so that when you place the slice on a glass slide, light from the microscope can pass right through the tissue to your eye.
Finally, we reach step 4, staining. Because natural, incredibly thin slices of tissue are practically transparent, they lack inherent contrast. They're basically invisible. So, they must be stained. The most famous and widely used combination in all of histology is the H&E stain. Hematocostilin paints the cell nuclei a vivid blue or purple color, while eosin dyes the cytoplasm and the extracellular matrix a bright pink or red. This essentially paints the microscopic world in these incredibly vivid colors, allowing anatomists and specifically medical pathologists to clearly identify the structure and spot the difference between healthy and diseased cells.
Section 5. Future of microscopic anatomy. Looking inward without the scalpel.
Let's look at where the field of histology is heading next. We've just talked about how tissues are historically extracted, fixed in wax, and sliced up. But the future? The future is taking a radically different approach. This is the concept of in vivo histology. Instead of surgically removing tissue, fixing it in chemicals, slicing it and dyeing it, modern medicine is pushing towards examining our microscopic architecture non-invasively, right inside the living body. By pushing the boundaries of high-resolution MRI technologies, scientists are working on ways to gather this crucial microscopic data in real time, completely intact, while the patient is simply lying in a scanner. It's a real game changer.
And that is the absolute incredible frontier of microscopic anatomy. We've gone from the 17th century microscopes of Marcello Malpighi to being on the verge of mapping microscopic living tissue with magnets and radio waves. It leaves us with a really profound question to ponder. If we can view the very fabric of our bodies in real-time, living action, how drastically will that change the future of diagnosis, medicine, and our fundamental understanding of human life? Thank you so much for joining me for this explainer. Keep wondering, keep learning, and I'll catch you next time.